All right. Hello, everybody. Thank you for checking this video out. Today we're going to go over DNA structure and some basics about DNA. Hopefully this is a topic that you know something about. Again, a lot of students in the past have seen some of this before, maybe some more detail this year than what you've heard in the past, but the hope and the goal here is to help everybody, whether or not you've seen it before. But my guess is many of you have heard at least of some of this stuff. So we will see. But again, here when we're talking about what we are looking for in our learning targets, okay, you want to describe the components and characteristics of life and how they apply to all living things is the main target. But here we're looking at differentiating the types of nucleic acids. So we talked a little bit about the macromolecules at the beginning of the year. We'll swing back to that later. But here today in this lesson, our goal is to discuss one particular type of macromolecule called a nucleic acid, and specifically DNA, which is deoxyribonucleic acid. Okay, so we're going to talk about DNA structure here today. So what is DNA? Obviously, you've heard of it before. Okay, again, it stands for deoxyribonucleic acid, and you might see here in a second why it got that name. But another way of thinking about this is that DNA is the molecule of heredity, is what we call it. It contains all the information, which in this case means genetic code that makes you, right? In other words, it creates the proteins that are the physical, tangible parts of your body. So we call the building block of DNA, we call that a nucleotide. Okay, another name for this is a monomer, and we'll talk about monomers when it comes to other macromolecules later. But for now, please take note of the word nucleotide, as is an important one. And each nucleotide contains three main parts, and this is where we get the name DNA from, right? That full name. So the sugar of DNA is called deoxyribose, hence the name deoxyribonucleic acid, right? So the sugar is what gives the first letter of the name. Okay. Another component of a nucleotide is what we call the phosphate group, right? Or a phosphate. That consists of one phosphorus atom bonded with four oxygen atoms. You don't have to worry too much about the specifics there of the phosphate group, other than knowing that it's a phosphate group. Okay, but that is what it consists of, just so you know. And then the third part is the one that you're most familiar with, right? These are called the nitrogen bases, or bases for short, and there are four of them. They are abbreviated with the letters of the first letter of their name, right? So A, T, C, G are the four nitrogen bases, and so one of them will be found on a nucleotide. All right, here's one picture of a nucleotide, just so you have some background there. Here is another one. So what you should notice, again, when we talk about macromolecules, they are essentially just repeating chains of some sort of base structure. And for DNA, that base structure is symbolized right here in this little chunk. So we have a P, which is the phosphate group, or sometimes called phosphoric acid. Don't be too worried about the difference there. You can call it either one. Most people will call it a phosphate group. Then we have the sugar. Okay, and so notice that the phosphate and the sugar are connected here in one direction. And then in the other direction to the sugar, we have what we call the base, or the nitrogenous base, or organic base. Okay, don't worry about these words here yet. But if you look here, this PSB, this is just repeated over and over again. Now remember, this is a single chain of DNA. DNA does have two strands connected in the middle that makes it look like a ladder, but in essence, DNA is just a repeat of these little phosphate sugar base parts on both sides, okay? And this is called a nucleotide again. So remember, another way of conceptualizing DNA is long strands of nucleotides bonded together. All right. Another name you have heard is the double helix, right? And so DNA is called a double helix because it looks like a ladder that has been twisted into a spiral, right? So again, the double is because it's two strands, one on each side. So this left side of the molecule is one strand, and this right side is another strand. So there's the double part. And then helix is just another word for spherical in this sense, right? That helix shape. So that's where the name comes from. So if you untwist the double helix, as you might call it, DNA looks like a ladder, right? And so we will distinguish DNA by the sides of the ladder, which are these sugar and phosphate repeating edges. And then we have the middle of the ladder, or otherwise known as the rungs. And the rungs are where the bases connect. And this, like I said, is the most interesting part of the molecule. This is where we spend most of our time talking about. So the middle is the bases. The sides of the ladder are the sugar and phosphate repeating. So how did the strands stay together? Well, one thing to know, and again, this is another one of those just like classic things about DNA that you absolutely have to understand, okay? Even though there are 4 letters, only 2 of them bond together. So in other words, every time you see an A on one side of the DNA chain, you will always see a T on the other side. So if I go back one slide here real quick, notice here at the top, I've got an A bonded to a T, an A on this side bonded to a T, an A on this side bonded to a T. So A's and T's always go together. And then likewise, G's and C's always go together. Okay, so I'll go back here to the previous slide. Now, some of you may wonder why this is the case. All right, and I would say this is kind of one of those strive for 5 level understandings, but it is potentially worth writing down. The reason why A's and T's go together is because DNA is held in the middle by a chemical bond known as a hydrogen bond, and both adenine and thymine, which is what A and T stand for, they want to form 2 hydrogen bonds, while the C and the G, cytosine and guanine, they want to form 3 hydrogen bonds. So this is why A's and T's always go together and C's and G's go together. So how can you remember this? Because again, as I said, really, really important that you know what goes with what. Well, one way of thinking about this, right? Again, this may not help everybody, but one way of thinking about this is that A's and T's, if you capitalize them, they all use straight lines. So the 2 letters that use straight lines go together, and then to write a C and a G, you for both of those you need curved lines, right? Like this big swooping line. So the one with this big curvy line goes together as well, right? So the straight line ones go together and the curved lines ones go together. Now, there is a term, another term that we introduce here that refers to the fact that we know what letter bonds to what letter. In other words, it's predictable. If I give you one side of a DNA molecule, like I did down here at the bottom, you can tell me what goes in below. This is called complementary, and we would call these complementary strands to be specific. So as we said, they are called complementary to each other. This does not mean they like to tell each other nice things, right? That's complement with an I. Complement with an E, kind of like in math. If you remember, complementary angles are 2 angles that equal 90 degrees. So if I give you one of the angles, right, if I tell you one angle is 50 degrees, you already know the other angle because complementary angles always equal 90, right? So if I tell you one is 50, the other one has to be 40. Same thing here in DNA strands, right? If I tell you one side of the DNA molecule, you know the other side. So what I want you to do now is pause this video just for a second and try to tell me the letters that would go opposite these letters here. Okay? Hopefully you have unpaused it after you've given this a shot. So again, don't go any further than right now if you haven't tried this, but I want you to try to write your own complementary strand here. But we'll go ahead and show you the results. Okay, this is what you should have written down on your notes. So notice again, an A is on one side, a T is always on the other. A is on one side, T is on the other. Same thing goes in opposite. If a T is on one side, an A is on the other. The other pair that we have is C's and G's. So a C is on one side, a G is on the other, a G is on one side, C is on the other. Okay. Now, again, as I said, terminology here is not as important. However, some people are going to hear more about this, and there is a way to kind of use these vocab words to demonstrate some more advanced knowledge, right? So something to keep in mind is that whenever we see a bond, it's always going to be between one, what we call a purine, and one pyrimidine. So you'll notice the purines have 2 of these ring-like shapes in their chemistry, right, in their chemical makeup. So adenine and guanine are the purines, and then cytosine and thymine are the pyrimidines, which only has 1 of those ring shapes. Don't worry about uracil down at the bottom. That pertains to RNA, which we're not going to get into yet. So something to think about, okay, is that if you are looking to maybe get into that more advanced understanding, think about the fact that purines and pyrimidines are always bonded together. And once you know a bit more about some of their relationships, you'll be able to tell just by knowing like one of the like the percentage of the DNA molecule that is a guanine, for example. You'll be able to know what all the three other bases represent because of, like I said, this predictability because of the complementary nature of their strands. All right. So, like I was just saying, since an A always pairs with a T, you always have a purine, which are those two-ringed molecules paired with a pyrimidine, which is a one-ringed molecule. Okay. Now, a really, really, really important aspect that I'm really gonna ask for you to write into your notes right now. Okay, because even though this is something you can derive or like figure out on your own, I just want to go ahead and say it. So if we go back a couple of slides here to this one here. So the strands stay together because A's always bond with T's and C's always bond with G's. So what I would ask you to add to your notes, and you can do this at the end or you can do it back where you wrote this original information, because A's always bond with T's and C's bond with G's, the same amount. So for example, if a strand of DNA is 20% A, it will also be 20% T, right? So hopefully that makes sense, because again, if an A is always found bonded to a T, well then however many A's I have, I'm going to have the same number of T's. The same is true for C's and G's, right? So try this out, okay? If I have 20% of my DNA that's adenine or A, then I know 20% will always also be thymine. But what if I said to you, what about C? How much cytosine would be in there? Well, again, if it's not obvious, if there's 20% A and 20% T, that means 40% of the molecule has been represented, right? So that leaves 60% that is unknown. So if I know that these two are going to be equal and between them, C and G represent 60% of the molecule, right? Because 40% was taken up by the A and the T, then I know that 30% of the DNA molecule will be cytosine, 30% will be guanine, right? So again, be very careful, because on a future test, I might say something like, okay, a DNA strand has, 15% thymine. Tell me the percentage of all of the other bases. You should be able to determine that knowing this. Okay. So hopefully that made sense. If not, let me know. But thank you for watching. And if you have any questions, please talk to me either by email or at the next Google Meets. All right. Have a great day, folks.